13 in biopharmaceutics, and the topic is Introduction to Protein Binding. Outline for today's lecture. In this lecture, we will come across with the first topic, that is what is protein binding, and then various types of protein binding, in which we will study about irreversible protein binding and irreversible protein binding. So what is protein binding? As we know that there are various endogenous, exogenous substances in the body that are destined to interact with various proteins depending upon their nature and affinity. Now, you know that what is meant by endogenous substance. Endogenous substance is the one which is found inside the body naturally, and it is not compulsory to be administered outside. So while what is exogenous substance, which doesn't which is not found naturally within the body, but it is administered from the outside, or it is any foreign substance which enters the body. So when it enters the body, what happens that it starts to interact with various proteins, no matter whether it is endogenous substance or exogenous substance. So proteins might be within the blood, or these could be various tissues. So any endogenous or exogenous substance is destined to interact with these proteins, and basically this interaction is the one which we call protein binding, because this interaction results in development of a complex, and that complex is a protein bound complex, and the process is called protein binding. Drug protein binding, as we just discussed, that it is basically a macro complex, and when drugs as exogenous substance enter the body and interact with the proteins, they make a complex which is called basically a drug macromolecule complex, because protein is something which is a macromolecule, and these proteins could be found within the blood, or these could be found within the tissues. In the former case, these would be called the plasma proteins. In the later case, it is called tissue proteins, and the examples are the macromolecules such as melanin and DNA. But basically, these are the proteins which result in irreversible protein binding, but there are various tissue proteins which might result in reversible protein binding as well. So types of protein binding. Protein binding can be reversible or irreversible one. The reversible protein binding is the one in which usually the drug binds with plasma protein, or if other tissue proteins it binds. Usually it is the reversible one, because the bonding is not that strong, and the drug gets free. Drug can get free once it is bound after that. While in case of irreversible process, what happens that once the drug gets attached to any protein, and usually these are the tissue proteins, or like the melanin or the DNA, so once it gets bound, it is usually for a very prolonged period of time, and the binding is a very strong chemical bonding. Usually it is a covalent bond. Irreversible protein binding. Irreversible drug protein binding is usually a result of chemical activation of the drug. Drug gets chemically activated, and that is an activated intermediate, and in that form it is very easy for the drug to get attached to any protein with the help of strong bondings, and this bonding is a chemical bonding, and that is known as covalent bond. It is a strong one, and that's why once this bonding happens, it is very difficult for the drug to get free and reverse the process of binding, and so this sort of protein binding is called irreversible one. So what is the order in irreversible protein binding? The drug will first convert it into its reactive intermediate, and then that reactive intermediate will react with the proteins, and their reaction will result in drug protein complex formation. Uh, the irreversible protein binding might result in toxicity. Irreversible drug binding accounts for certain types of drug toxicity that may occur over a long time period. As we just discussed, that the drug, when converts into its reactive intermediate, starts to interact with the tissues, and this results in a strong covalent bond formation that leads to the irreversible protein binding, and this could be over a prolonged period of time, and the reactive intermediate, when once gets attached to the proteins, it is the same phenomena as a chemical reaction is happening on the surface or within that tissue. So over a prolonged period of time, this might result in into some sort of a big damage to that tissue, like as in the case of chemical carcinogenesis. Many chemicals, after forming reactive intermediates in the body, get attached to proteins, and this results in chemical carcinogenesis. And within a relatively short time period, as in case of drugs that form reactive chemical intermediate, paracetamol, liver toxicity is a popular scenario as well, as we know that paracetamol, usually in the normal scenario, what happens that paracetamol or many other drugs, they are forming their metabolites ready to be excreted, which are more polarized, but let's suppose due to some reason there is glutathione deficiency within the liver, which might be due to the overdose, or it could be because of some pathological condition, and the drug won't find its usual process to happen to convert its reactive intermediate into the metabolite which is ready to be excreted. Now that reactive intermediate is not able to get excreted in the form of any other polarized metabolite, so that reactive intermediate will start to interact with the tissues because of glutathione deficiency. So that result in immediate sort of liver toxicity, and which is very common in case of paracetamol poisoning or toxicity. The hepatotoxicity of high doses of acetaminophen is due to formation of reactive metabolite intermediate that interact with liver proteins. We just discussed the same thing in previous slide. Reversible protein binding. So most of the drugs choose reversible protein binding when it comes to their binding with proteins, and it means that while doing that, drugs are not undergoing the formation of reactive intermediates. Neither they are forming strong bonds, covalent bonds, but they are engaged with forming weaker bonds, which are also called the hydrogen bonds or the van der Waals forces. These are weaker bonds, can be broken at any time point easily, and the drug can detach from the protein at any later stage. So so that's why the reversible protein binding is of great importance as far as pharmacokinetics is concerned, because pharmacokinetics is mainly concerned with movement of the drug from uh, within the body, or its distribution, its elimination, metabolism, whatever is, it is basically movement of drug within the body, how that is happening. And what the drug is doing to the body is what you call uh, pharmacodynamic, and pharmacokinetics is what is happening to the drug within the body. So that is all what you call the movement of the drug within the body kinetics. And when it comes to pharmacokinetics, reversible drug protein binding is of great importance, because at any stage later on, drug gets detached, distributed, and gets attached to the receptors and produce a certain pharmacological response then. So, reversible drug protein binding implies that drug binds the proteins with weaker chemical bonds, such as hydrogen bonds or van der Waals forces. The amino acids that compose the protein chains have hydroxyl, carboxyl, or other sites available for reversible drug interactions. So, when we say that uh, why such drugs prefer, or so many drugs prefer to undergo reversible protein binding, how is that possible? So basically, whenever the drug is not making a reactive intermediate, the usual sites for interaction, for possible interaction with proteins, are hydroxyl, carboxyl, or other sites, which are the hallmark characteristic of the amino acid groups within the proteins. They are usually available for um, drug interaction with the proteins, which is a reversible sort of. So hydroxyl, carboxyl, or other sites are important sites for developing weaker bonds, reversible bonds. And this you can better see in this diagram of amino acid structure. And here you can see that within a carboxyl group, hydroxyl group availability makes it a good candidate for forming hydrogen bondings or the weak van der Waals forces development. So that is the main reason that availability of such groups makes the possibility of reversible interaction of drugs with proteins so possible and frequently happening. So what would be the impact of protein binding on the volume of distribution? So protein bound drug is a large complex that cannot easily transfer cell or even capillary membranes, and therefore has a restricted distribution. So what does it mean? That a volume of distribution would be the first pharmacokinetic parameter, which would be get affected by protein binding, drugs protein binding, and drug won't be in a position to get distributed until unless it is in bound form with the protein, because it is making a large complex which cannot easily traverse the capillary membranes and cannot go to the other side, or you can say it cannot be easily distributed towards the other tissues or the peripheral areas of the body, and so it makes the drug to be restricted within the blood circulation. So volume of distribution would be restricted, limited for the drugs specifically which are highly protein bound and lesser lipophilic as well, because we already discussed that if the drugs are highly lipophilic, at least when they get free, they have this tendency to get distributed to, or they could show a propensity to get attached to the uh, lipids or fats tissue within the body or many other deeper areas within the body. But usually the drugs which are hydrophilic and highly protein bound as well, they are highly limited or restricted within the blood circulation, and it is very hard for them to go and get distributed within the greater or the deeper areas in the peripheral areas. So such drugs have a limited volume of distribution. Okay, students, so lecture is still continued, and in upcoming lecture we will discuss about various types of proteins which are involved in protein binding with the drugs. Thanks.